All right, welcome back, guys. This is Biological Molecules Part 2. In the first part, we talked about the basics of reactions that link molecules together, and we talked about carbohydrates. This video is going to cover lipids. And lipids are a little bit different than other biological molecules in that they don't really form true polymers. So unlike glucose, where you link lots and lots of molecules together into a long chain, this is a little bit different. There are a few different types of lipid molecules that we assemble, but they don't really look like the others in the sense that we don't link lipid after together on a really long chain the way that we do with carbohydrates or amino acids to make proteins. And so in this case, what we're doing is we're taking a backbone molecule, glycerol, which contains 3 carbons, and each of those carbons has an OH on it. Right? An OH is an alcohol group, and so technically glycerol is an alcohol, and what we're going to do is we're going to use that hydrogen to combine it with an OH that's on the end of this molecule called a fatty acid. All right, so a fatty acid is just exactly that. You have a carbon double bonded to an oxygen and then single bonded to an OH, and then a chain of carbons and hydrogens. This chain is the part that's variable. It could be as short as 4 carbons long or as long as 30 or 40 something. So the number of carbons is wildly variable, but that's the pattern that you're going to see, right? And just like with carbohydrates, if we want to build molecules together, we need dehydration synthesis. So we're going to take that H and this OH, combine them together and remove a water molecule. That's the dehydration part. And we end up then with a linkage. Right? This triacylglycerol, because we have 1, 2, 3 carbons on the glycerol, so we can attach 3 fatty acid chains. And this is the perfect molecule for energy storage. Right? The reason for that is they are incredibly nonpolar. Right? Remember, anytime you see a bunch of carbon-hydrogen bonds together, they have similar electronegativities. There's no oxygen or nitrogen anywhere in that chain. And so remember, nonpolar effectively means hydrophobic. These things do not like water. So this is the exact reason why if you go into your kitchen and you pour some cooking oil into a jar of water, the oil will rise to the top and they'll separate. We call that the hydrophobic exclusion effect. Water molecules do not like all that nonpolar carbon. This is great for plants and animals that want to store energy because you can take a bunch of triglycerides, pack them tightly together, and you don't have to waste energy carrying around a bunch of water to dissolve those triglycerides in. So this is what human adipose tissue is made from. This is what every mammal or animal uses to store fat. It's even what most plants use to store fat. So it's a fantastic way to store energy and save it for later. All right, within those fats, you have a few different kinds. The term saturated fatty acid just means that the carbons in that chain are saturated with hydrogens. And if you remember from your basic chemistry, carbon always forms 4 bonds, right, to fill its electron shells. And so every one of these carbons in a saturated fat is bound to 2 other carbons and 2 hydrogens. So when we say this is a saturated fat, it really just means single bonds all the way down. You have saturated the maximum number of hydrogens that it can possibly have, right? As soon as 2 of those carbons form a double bond, we have to take away 2 hydrogens, right? If you notice up here, every carbon had 2 hydrogens attached to it. If we add a double bond, now each of these carbons only has room for 1 hydrogen because it can only form 4 bonds total. And we have 1, 2, 3, 4. These hydrogens down here had to get deleted. Okay, so we say that this is an unsaturated fat because it has less than the maximum possible number of hydrogens that it could have. Now, if you watched my video on isomers in the introduction to chemistry, you'll remember that if both carbons after the double bond are pointing the same direction, like these are, we say that that is the cis isomer. So this is cis oleic acid because the chain is bent. They're both pointing the same direction. If you see trans oleic acid, that just means that the carbons are pointing opposite directions, right? One up, one down. That makes the chain look a little bit more straight. And so trans unsaturated fatty acids actually behave a bit more similarly to saturated fats, right? The reason that in your kitchen, saturated fats tend to be solid at room temperature and unsaturated fats tend to be liquid is that these solid saturated fatty acid chains, they stack and pack really easily because the chains are flat, they pack tightly. Like imagine toothpicks in a box. For the unsaturated fats, imagine taking all those toothpicks and bending them a little bit to break them so that they form a bent angle like this. They wouldn't pack neatly in the box, and so they move around more easily and they stay liquid. Trans fats behave kind of like the saturated fats. That's why food manufacturers for a long time artificially converted cis fats into trans because they wanted to give their food a more solid feel and not have it be all runny and liquidy. The problem is it turns out that humans don't have the enzymes necessary to handle trans fats well, and they are enormously associated with heart disease and heart attack because they form plaques and cause inflammation inside blood vessels. Right? So for these reasons, the properties of fats, whether they're saturated, unsaturated, cis or trans, makes a big deal for how cells handle those fats and how membranes are formed, which happens to be where we're going next with this discussion. Okay, so molecule type 1, triglycerides, fantastic for energy storage. Not very good for forming membranes because triglycerides, if you mix them with water, generally just form a big blob. These molecules, phospholipids, are ideal for membrane formation. And the reason for that is the phosphate up top, this PO4, or the phosphorus and 4 oxygens, has a negative charge and a bunch of oxygens. This makes it incredibly polar, right? And remember, polar means hydrophilic, right? If a molecule is hydrophilic and it likes water, it's going to preferentially want to interact with water, right? The tails, on the other end, are very hydrophobic, just like in the triglycerides that we saw before. And so this phospholipid becomes a molecule that's something called amphipathic. It just means that it has one end that's hydrophilic and it has another end that's hydrophobic, right? Within those fatty acid tails, they can either be saturated or unsaturated. This picture shows one of each, but that's not a rule. They could both be saturated or both be unsaturated. It kind of depends on what type of organism you're looking at and what they need from their cell membrane. Right. So what happens in a biological setting? If you take phospholipids and you mix them with water, like pretend that these were existing inside a container of water or inside of a living cell, you would expect water to be found on both sides of the structure. And so this phospholipid bilayer forms spontaneously. There's no enzyme that has to make it happen. Nothing in the cell has to tell these phospholipids to turn into a membrane because the heads, right, these are your phosphate heads out here, they're hydrophilic and they like water, so they orient this way towards the water outside the cell. And these phosphate heads on the other side, also hydrophilic, they like orienting themselves this way to also interact with water. The tails are all hydrophobic and they prefer to orient towards each other because hydrophobic things like to interact with other hydrophobic molecules. And so the bilayer forms spontaneously because that way everyone's happy, right? The lipid tails get to contact each other here. The phosphates all get to touch water pointing on both sides. And so if you take phospholipids, put them in a jar and shake it up, you will naturally get these membranes forming, which is perfect for cells because they don't have to do anything to make their membranes form. They just start producing phospholipids and they'll spontaneously take on that structure. Okay, so recap, we've got two major types of lipid molecules that we're dealing with, right? We've got triglycerides, which are almost entirely nonpolar and hydrophobic, that are very good for energy storage because they form big, dense droplets that can be stored as adipose or fat tissue and then saved for later. And separately from that, you have phospholipids, which still use fatty acid tails, but remember have phosphate heads to make one end hydrophilic. That way they spontaneously form membranes. Okay. The last category of molecule that you should be aware of that fits into this lipid category is cholesterol. And cholesterol doesn't really look like fatty acids in the sense that it's not just a long chain of carbon and hydrogen. cholesterol takes on a ring structure. So remember, when you see these rings drawn, every corner of the ring is a carbon. They're just not drawing all the C's in, right, to make it easier to follow. And so this cholesterol molecule, I know cholesterol gets a bad rap. It's associated with heart disease in some ways, but it's actually a crucially important biological molecule that you couldn't live without. And the reason for that is cholesterol is an important part of cell membranes, and it's also used to make steroid hormones. So this term steroids really just means any molecule that's made from cholesterol. So your list of steroids includes important hormones like testosterone, estrogen, cortisol that you see in the picture down to the right, other hormones like aldosterone, many, many things that you'll see if you eventually move on to a physiology class. These make very efficient hormones because they're lipids, they're lipid soluble, they can diffuse directly through cell membranes. And so when endocrine organs in a human or an animal make these hormones, they can actually very easily enter cells. They can also be absorbed through the skin. That's why when you go to a drugstore and buy cortisone cream, like an anti-itch cream, that cortisone is basically synthetic cortisol. It can diffuse directly through your skin, get into the blood, and help reduce inflammation because it's lipid soluble and can move directly through those membranes. All right. Okay, that's the end of our primer on lipids. Follow along to the next video, which will cover amino acids and how they're used to build proteins. Make sure you follow the playlist. I've got these videos organized in order as they go through the book, and subscribe to the channel so new ones pop up. We can help you all the way throughout your class.